So a heartbeat, a single heart contraction. The entire heart contracts in a series, first the atria, then the ventricles. If we look at cardiac physiology, there are two types of muscle cells. You've got the conducting system cells and the contractile cells. The cardiac cycle begins with an action potential at the sinoatrial node, or the SA node, and this is transmitted through the conducting system, and it produces an action potential in cardiac muscle cells, the contractile cells. We as health professionals can monitor this cardiac cycle electronically by an ECG or an EKG. The electrical events of the cardiac cycle can be recorded, and diagnoses can be made based on that. So the conducting system, it's a specialized system of cardiac muscle cells. They initiate and distribute electrical impulses that stimulate contraction. The cardiac muscle cells have automaticity, which means that they can contract automatically. So if we look at the structures of the conducting system, you've got the sinoatrial node in the wall of the right atrium, the atrioventricular node, which is the junction between the atria and the ventricles, and then you've got the conducting cells throughout the myocardium. The conducting cells interconnect at the sinoatrial and the AV node, and then they distribute the stimulus through the myocardium. In the atrium, there is what is called internodal pathways, and in the ventricles, there is the AV bundles and the bundle branches. So I'll show you a diagram in just a moment. The pre-potential is what is called the pacemaker potential. This is the resting potential of the conducting cells, and it gradually depolarizes towards threshold. So the SA node depolarizes first, establishing the heart rate. So that is called, the sinoatrial node is often referred to as the pacemaker of the heart. So this is the diagram of what we've just been talking about. You can see here the purple loops that is on the surface of the heart. So of course this is all electrical conductivity of the heart. You can't actually see it if you were to dissect the heart, but research has been done to show that this is the way the heart is actually electrically stimulated. So in the right atrium, you have the location of the sinoatrial node. At the base of the right atrium, at the atrioventricular junction, you have the AV node. And then going down through the septum, which separates the left and right ventricles, you have the AV bundle, and then that branches off into the branch bundles and then into the Purkinje fibers. You will be expected to know the names of the conducting system of the heart and the location of them. So the changes in the potential membranes, so they show you how the actual pacemaker of the heart works. So the sinoatrial node is established at a heart rate of around 72 beats per minute. So the sinoatrial node generates between 80 and 100 action potentials per minute, and parasympathetic stimulation slows the heart rate down. The AV node can generate between 40 and 60 action potentials per minute. So the sinoatrial node is in the posterior wall of the right atrium, and it contains the pacemaker cells, and it's connected to the AV node by the internodal pathway, and it begins the atrial activation. So this is step one. So here you can see at the top of the right atrium, the sinoatrial node, and at the bottom you can see the AV node. So this is when it is time 0. The atrioventricular node is in the floor of the right atrium, just as I said, and it receives the impulse from the sinoatrial node. This impulse is delayed to allow for blood to continue to drain from the atria down into the ventricular, and also it allows it to be delayed just for milliseconds so that the electrical impulse can spread over the left atrium. So here we've got the stimulation spreading across the atrial surfaces of the right atrium and beginning across the left atrium. So time elapsed is about 50 milliseconds. There is a 100 millisecond delay at the right atrium, so this allows the blood to move down into the ventricles. And now the AV bundle has picked up the electrical stimulation, and that's going to be sending it down through the bundle branches. It carries it to the left and right bundle branches, and then on to the Purkinje fibers. And then the moderator band, which conducts through to the piliary muscles. So here you can see the impulse travels down the intraventricular septum within the AV bundle and the bundle branches to the Purkinje fibers via the moderator band to the piliary muscles of the right ventricle. The Purkinje fibers distribute the impulses through the ventricles. The atrial contraction is complete, and now the ventricular contraction begins. So you can see here the purple shading is moving, the conduction is moving up through the ventricles, the myocardium, and the atrial contraction is completed, and now the ventricular contraction begins. So if you have abnormal pacemakers, the sinoatrial node, you can have what is called bradycardia, slower than normal heart rate, or tachycardia, faster than normal heart rate. Or sometimes you can have what is called ectopic beats. These are or an ectopic pacemaker, where you get abnormal cells around the atrium which start sending off their own electrical stimulation. This can be a problem because it can affect cardiac output. As we said before, the ECG is a recording of the electrical events of the heart, and you will learn how to place the electrodes and how to record and interpret at a basic level the electrocardiogram. So here we've got an older gentleman lying down, and he's having his ECG being taken. It's really important that the person lies still, doesn't talk, so that there won't be too many vibrations or artifacts appearing on the cardiac tracing that is actually being made in the machine that you can just see at the top of the picture. Features of the ECG, we start with the P wave, which represents atria depolarization, the QRS complex, which shows you ventricular depolarization, and the T wave, which shows you ventricular repolarization. The PR interval is the start of atrial depolarization to the start of the QRS complex, and the QT interval is from ventricular depolarization to ventricular repolarization. All that sounds like words, so let's have a look at it on an actual ECG tracing. So here you can see the top bit is a strip of ECG paper, and each square represents 10 milliseconds. So you can count up the squares to work out the rate of the heartbeat. And here below that we've got the heart tracing with all of the intervals labelled. So again, the P wave is when the atria depolarization is occurring. The QRS is when you've got ventricular depolarization and ventricular contraction, and then you've got the T wave when you've got the ventriculars repolarizing. Behind the QRS is where you would see the atria repolarizing, but because the QRS is so powerful, you don't actually see that on the tracing. So here are some abnormal rhythms. You wouldn't be expected to be able to pick up these. All we're asking for you is to be able to notice what is normal sinus rhythm. You can imagine the bottom one, ventricular fibrillation, where the ventricles are not contracting at all, that the person would be suddenly dropping to the ground and becoming unconscious because there's no cardiac output. There would be no blood supply going to the brain. So within the conducting system, there needs to be a period of rest, what is called the refractory period. So there's two, one is absolute and one is relative. So the absolute one is longer and the cardiac muscles can't respond, and then there is a shorter one when they can respond depending upon the amount of stimuli. The amount of stimulation and the quickness of the response depends upon the presence of calcium ions. So the contraction of the cardiac muscle is produced by an increase in calcium ions and concentration around the myofibrils. So you can see that calcium as an ion and an electrolyte is vital in the body, as is potassium. The energy for cardiac contractions, it is aerobic energy from the mitochondria. We saw in lecture 1 when we saw the the cardiac myofibrils, that there are many mitochondria breaking down fatty acids and glucose. And then of course you've got circulating oxygen with circulating hemoglobin from the red blood cells, and the cardiac muscle can also store a certain amount of oxygen in the myoglobin.